So, what we're going to be talking about tonight is really what methylation is. You're also going to be learning how methylation impacts autoimmune diseases. We're going to talk about some common methylation issues in autoimmune disease, and then also how to support methylation for autoimmune health. And there will also be a live Q&A at the end. Now, the session is being recorded, so when it comes to that, if you'd rather not appear on the recording, just type your questions into the chat and I'll try and make sure I answer everything. Otherwise, just feel free to ask questions as you please. I'm just going to see if I can change this view so that you can see me, but it doesn't look like I can. So let's just stick with where we are. So let's have a quick look at methylation and what it actually is. So methylation is basically it's an enzymatic process that requires cofactors like folate, B12, B6, and SAMe. And what it actually facilitates, or what it does, it's the transfer of what's called a methyl group. So this is a compound with the chemical structure of CH3. So carbon, C is carbon and H is hydrogen. And it puts it onto DNA, protein, and other molecules, and it catalyzes or sort of fuels different reactions. And so what you can see here, this is a diagram that shows a very simplified version of the methylation cycle. So it's not just one reaction, it's a series of different reactions that all sort of happen as part of a process. And so, if we start here, so what I've actually got here in this diagram, in red here, these are the genes that actually produce the reactions within the cycle. And then these are the enzymes that are produced by the genes. So we've got it sort of going around here. So what you actually have is methionine comes in. So this is an amino acid. And it comes in, it sort of goes through this cycle through a few different forms until we get to homocysteine, which we'll talk about a little bit later, and which some of you might have heard of as a good measure of how methylation is working. And so it goes through there, it comes through these two genes, MTR and MTRR, and it comes round through, so it gets to tetrahydrofolate here. We've got 10-formyl tetrahydrofolate, and then it goes through another couple of versions of folate, basically, and some more genes there. And then we get to MTHFR, or motherfucker, as it's sometimes called, which again, we will talk about a little bit later. Comes back round, and then it can either go back round through this cycle or it can come over here through this cycle, sort of depending on what's going nutritionally, and also where it's needed. And you can see that there are also what we call sort of redundancy loops. So you've got two sort of alternative pathways here. So you've got the SHMT pathway with B4, and then you've also got this pathway here where it goes through BHMT. And again, we'll talk a little bit more. We won't talk that much about SHMT. We will talk a tiny bit about BHMT later. But that's basically what methylation is. It's a cycle. So it's not just one chemical reaction. It's a cycle that relies on a whole series of chemical reactions to work well and to really sort of do what it needs to do. And that means that it relies on a number of different genes that produce a number of different enzymes that actually facilitate all of those different reactions that it does. So what is methylation involved in? And you can see from this diagram here that it's actually involved in quite a lot of things. Now, this is necessarily a very simplified diagram. I don't think I could possibly manage to actually make a diagram as a PowerPoint that's got everything involved in there. It's involved in around 200 or over 200 different processes in the body. So it's a really, really important process and it governs a lot of different things within the body. So it's really important. But just to give you a bit of an idea, what we've got here is, creatine is produced through methylation. Some of you might take creatine as a supplement. Some of you might have heard of it as something that people who go to the gym take as a supplement. But creatine is really important for energy production. So, some people with autoimmune disease who feel really tired can feel great when they take creatine because it's so closely linked to methylation. It's dependent on methylation, and people with autoimmune diseases often struggle with methylation. You've also got it doing things like making neurotransmitters. So these are the chemical messengers used by our nervous system and the brain to communicate. And so if you have symptoms like brain fog, if you find that you're really forgetful and those kind of neurological sort of just not quite up to speed, those kinds of symptoms, that, can potentially be something related to methylation. Not always, but it can be. We've also got DNMTs here, and they contribute to epigenetic regulation. So, epigenetic regulation is the process by which we turn certain genes on and off. And if you've been following me for a while or you attended the last live seminar that I did, then you probably know this already, but I'll just recap for people who don't know. Within the human genome, so that's our entire DNA sequence, we have got about 10 to 15% of genes that are really quite fixed once we go through what's called developmental periods. So these are periods of development where those genes are kind of triggered to turn on, and we then end up with, say, blue eyes or brown eyes or green eyes, or we end up a certain height or with a certain hair color, those kinds of things that tend to be more fixed. But then we have this whole swathe of other genes. So that's sort of 85 to 90% of our genes that actually change in terms of their expression throughout our lifetime. And that includes things like the genes involved in the immune system. So the vast majority of the genes involved in the immune system are actually turned on and off, including the ones that make antibodies. And we'll cover that in a little bit more detail soon. You've also got purine synthesis. And so purines are basically the base pairs that make up our DNA. So I won't go into too much detail about that, but they play a really important role in actually making DNA anytime we make new cells, and something called RNA, which fulfills a slightly different role to DNA, but it's kind of similar as well. And also making ATP, which is the energy that we use in our cells to sort of fuel everything that we do. And then this other really important thing here, we've got homocysteine converted to cystathione and then cysteine, and this fuels glutathione, sulfate, and taurine. Now, these are all detoxification pathways that take place within our cells and especially within the liver. So what methylation is also doing is it's allowing our body, or giving it the substrate, to be able to remove harmful substances from our body so that we're not harmed by them. And so these are some of the really important things that methylation is involved in. But as I've said, there's many, many more, but too many to go through here. So these are the key ones that I just want you to kind of remember. Now, why does methylation matter in autoimmune disease? And there's two reasons, or there's two kind of key characteristics of methylation that we see in autoimmune disease. And that's hypomethylation and there's hypermethylation. So, I mentioned briefly before that methylation plays a role in turning genes on and off so that we can be responsive to our environment. And there's a couple of different ways there's one way that it does this, and it's basically sort of regulating gene expression by putting a little chemical tag on your genes. And the gene that's been tagged and those around it will be silenced. So in hypomethylation, so hypo, think of it being not enough, we'll see that there are genes that are turned on that really should be silenced. And then hypermethylation, which is the opposite, we'll see too many genes that should be expressed are actually turned off. And both can cause problems. And guess what? Both are commonly observed in people with an autoimmune condition. And what's more, hypo or hypermethylation are often observed at the site under attack. So this is really, really important to remember. It's actually local to where your antibodies are attacking is often where that hypo or hypermethylation will be taking place. So it's going to be the organ or tissues being targeted by the antibodies. And as an example, I'm going to talk briefly about rheumatoid arthritis. So it's really well documented that hypomethylation can lead to aberrant gene expression in rheumatoid arthritis. So, and this is what this diagram is sort of here. So in rheumatoid arthritis, we've got synovial fibroblasts, and specifically fibroblasts like synoviocytes, which are these little cells here that you can see in this diagram. Hopefully you can see my mouse moving there. They actually become epigenetically reprogrammed. So hypomethylation of what's called the promoter region, you don't need to remember this, but hypomethylation in some of the key inflammatory genes in these fibroblasts like synoviocytes actually allow certain genes like interleukin 6 and tumor necrosis factor A and things like that. So you can see some of these here to actually be overexpressed. So what's really happened here is that the epigenetic regulation of those immune genes has become dysregulated. And what then happens is that those cells, these fibroblasts synoviocytes, actually create cytokines and chemokines that recruit and activate immune cells, including all of the ones that are involved in a normal immune response and an autoimmune response. So they release reactive oxygen species as well, and that increases the oxidative stress. So that's why you get pain from this. It's because of that, those oxidative species and the actual damage that they're causing to the cells and to that area. And then that fuels even more inflammation and even more tissue damage. Now, these cells, the fibroblasts like synoviocytes, will also secrete what's called matrix metalloproteinases, and they will degrade collagen and the extracellular matrix components. And they're quite invasive, which allows them to migrate into cartilage and bone, and that can lead to erosion and joint deformity. And then, these are really common features of rheumatoid arthritis. So, you know, these cells, fibroblasts like synoviocytes, don't actually orchestrate the antibody attack. That's actually mast cells that do that. But they're kind of the instigator of this whole cycle. And it's due to poor methylation. It's hypomethylation is really driving this whole response. And it leads to a really aggressive inflammation sort of perpetuating cycle. And we see this, in rheumatoid arthritis, but we see it in all of the different autoimmune diseases. The same kind of pattern where something local has become inflamed, and that then recruits the immune system, which then eventually can lead to the development of autoantibodies and the damage that's associated with them and with other inflammatory molecules. So really, really important, obviously, then that we have a look at methylation in autoimmune disease. Now, some of the common methylation issues in autoimmunity. So what we can really see is that people with autoimmunity have mutations in their methylation genes. So if you remember the cycle before, and the ones I think I have them in red, are the genes. And you can actually have variants of different genes that aren't the same as the original gene, and they maybe don't work quite as well. Sometimes you can have a mutation where it doesn't really affect the function that much, or it's very benign. It can be context dependent. But basically, these can cause issues in autoimmunity. But what I want to be really clear about here is that they don't actually cause autoimmunity. So these autoimmune diseases are not genetic, they are largely lifestyle diseases. So these genes, if you have a mutation in your methylation genes, it's a predisposing factor. They're like a weakness in your methylation cycle, which can be sort of pierced under certain conditions. And the most well-known mutations in the methylation cycle are, of course, MTHFR, also known as the motherfucker gene. So these genes are folate dependent. And they've got a really, really strong association with autoimmune diseases. So there are two variants that are particularly problematic. We've got A1298C and C677T. And so what does the mutation do? As an example, people with two copies of A1298C make up to 40% less of the enzyme, that MTHFR gene, which is also actually called MTHFR. So they make 40% less of this enzyme that gene is responsible for making, which is a pretty significant reduction. Now, people with two copies of the C677T actually make up to 70% less of the enzyme. That's 70. So this is a really important mutation to pay attention to. It can really mess up that methylation cycle, and particularly if there's a big need for methylation. So that's kind of the big one that will be the most commonly tested and that people will have heard the most about. But there are others as well. So another one is COMT. It's really closely linked to methylation. It's not actually there in the methylation cycle, but it sort of branches off it in a different direction. And it does a few different things, but the most impactful things in the context of autoimmune disease are that it leads to an inability to break down estrogen and dopamine at the same rate as people who don't have these mutations. Now, why is that a problem? Estrogen in high quantities can be really pro-inflammatory, particularly in the context of histamine, and if you have issues with histamine breakdown as well. But also, the inability to break down dopamine at the same rate is actually linked to ADHD. And so we've heard a lot about ADHD being the result of having not enough dopamine. Actually, having too much dopamine may be just as much of a cause. There's multiple causes of ADHD, but that can really be stressful for a lot of people. And the nervous system plays a huge role in regulating your immune system. But it can also lead to behaviors that may be sort of problematic for your health and for your oxidative balance within the body. So, having an excess of either estrogen or dopamine can be quite disruptive to immune function. Then we have got MTR, which is B12 dependent, and it really helps to move homocysteine through the two different cycles. So it facilitates that transfer between the two cycles and plays a big role in recycling homocysteine. We've got MTRR is another B12 dependent enzyme that works really closely with MTR. And mutations in both have been linked to an increased risk of an autoimmune condition. And then the other one that I think is really worth mentioning here is BHMT. And that was in that first part of the methylation cycle where it sort of provides an alternative pathway for homocysteine recycling with sort of the MAT genes. It relies on betaine, also known as trimethylglycine. So people with mutations in this gene are likely to have an increased need for betaine. Now, we don't necessarily see mutations in all of these in people with autoimmune diseases, but you will tend to see an increased prevalence compared to the general population in people with autoimmune disease. And what's really happening, or what I want to note here, is that just having these mutations doesn't mean you're going to get an autoimmune disease. As an example, I have two copies of the MTHFR C677T, plus mutations in BHMT, MTR, and MTRR. And while I did have an autoimmune condition, I don't have one anymore. I was able to reverse it and to bring myself back to health. And what happened before I got an autoimmune disease was I had lifestyle changes and I had stress and I had all sorts of things going on. So, if my life had sort of panned out differently, then I may never have gotten an autoimmune condition. So it's not a life sentence if you have these mutations. It just means that you have to be mindful of them and do what you can to support them. So, how to check if you have issues with methylation? And one way of doing it is through DNA analysis. So, this is where we actually look and see if you have mutations that affect methylation, which is something that I do. So I have a report that you can order on my website, and there's also a sample copy of it there, so you can see exactly what's involved. There's plenty of other providers around who also look at some of the methylation genes as well. And that's really going to tell you this is where your weaknesses are, this is what your predispositions are, and what you should do to support yourself nutritionally. But that really doesn't tell us everything we need to know. So we need to actually check functional biomarkers, which tells us what's happening at a functional level. My personal favorites for this are blood tests and urine testing, and I think they can be really, really valuable for this. And in an ideal world, every time I'm working with clients, I would get both blood and urine tests done to see not only what's available to the body, but also if it's actually using it. So urine testing is much less available than blood testing. It's really quite expensive, and your GP is very, very unlikely to order it. But if you're getting nowhere with what you're doing, it may be the next step that you need to take. If you've sort of exhausted all other avenues, is actually doing urine testing to see what metabolites you have. And that will tell you if you're using those nutrients that are present or not. I also use a different range for my blood biomarkers. So I don't use the lab ranges. I use an optimal range and really try and hone in on what's going to make a person feel better, not just what's going to stop them from being a complete mess and in disease state. And it can be really quite revealing to use those different markers. Now, the other thing that I would do to assess methylation is really to have a look at symptom patterns. So we can have a look at how issues with methylation might be manifesting for the client. So if they're experiencing an inability to concentrate, if they're experiencing fatigue, if they have ongoing histamine intolerance and these kinds of issues, we're going to have a look at them and do what we can to support them and constantly monitor them as well as we're trying different interventions and just making sure that anything we do doesn't actually make any of those symptoms worse, that we're heading towards continuous improvement. So how do you actually get that improvement? So what we really want to do is support methylation. And the most obvious thing is obviously through nutrition. So eating plenty of food with B vitamins, and then also, this is really, really important for everyone to note, avoiding folic acid at all costs. So this can be really tricky if you're in places like Australia, the US, or the UK, where commercial wheat is actually fortified with folic acid. So folic acid is the synthetic form of folate. It does not occur in nature. Let's just be really clear about that. It doesn't occur in nature, and our bodies don't know how to deal with it. I won't go into the history of how it came to be put into our wheat. But for now, just know that people, especially those with MTHFR mutations, find it really difficult to actually use folic acid, and it can actually stop you from being able to use natural bioavailable forms of folate. So if you have MTHFR mutations, and even if you don't, I would still suggest that you avoid commercial wheat and breads if you're in countries where it's fortified. So opt for things like long fermented sourdoughs, organic wheat products, or pasta from places like Italy where the flour isn't fortified with it. And then you need, on top of folate, natural folate, bioavailable folate. On top of that, you need a whole host of other nutrients, including other B vitamins. But then you've also got things like zinc, magnesium, iron, copper, betaine, vitamin C, selenium, and many more. Glycine is another one that would be really good to have in there as well. So that's an amino acid. Um, to really sort of support methylation and all of the different processes that it's involved in and sort of in the background. So what I would say with that though is it's really important not to, after this seminar, to just go out and run around and buy all of these different things in supplement form. And there's some really good reasons why. One is that you can actually have too much of a good thing. You can get side effects from having too much folate or too much B12 and too much B6. I think people in Australia will have heard about B6 recently. So you really want to make sure that if you're doing this, you are actually supporting yourself nutritionally first and foremost. So you're not getting super high doses of things. But then, if you are looking at supplementation, working with a health professional who can help you make good decisions about what you actually supplement with. Do you actually need these things? And that's really to avoid those side effects. Now, another thing, and this ties so closely into methylation, but I think it's so underappreciated, is vitamin D. And vitamin D actually regulates methylation. So we do not methylate if we don't have vitamin D. It's vital. And guess what? Almost everyone I work with has vitamin D deficiency. These are all people with autoimmune disease and other chronic health conditions, and all of them have got some form of vitamin D deficiency. So what does vitamin D do? So it's a hormone, it's an antioxidant, and it does all sorts of different things. But one of the things it does, which isn't talked about, is that role in methylation. And so it's really, really important for people with an autoimmune diseases. Now, something else to note here with vitamin D is that you might go and get your vitamin D tested with your doctor, and it will come back saying everything is fine. So, most people might not be aware of this, but the levels of different nutrients and different biomarkers that a lab can say is normal or not, it doesn't have to be updated in line with the most current research constantly. So what I'm seeing a lot with lab work that comes back is that most of these labs have not kept up to date with the most current research. And I've actually seen lab ranges saying that as low as 30 nanomoles per liter is okay when it's actually not. The most current consensus is, and so this is an expert consensus, these are people, researchers, medical professionals who study vitamin D and they're looking at it all the time. The current consensus is that 80 nmol per liter is the absolute minimum you need for health, and the optimal is anything above 100 nmol per liter. So if you're relying on lab ranges telling you that you've got enough vitamin D, please go back and have another look and get it checked. If you haven't had it checked recently, get it checked ASAP and then work with a functional, integrative, complementary, whatever type of practitioner who really kind of understands this and can help you get your vitamin D levels where they need to be. And I can guarantee that you're going to feel a lot better when you do this. Now, the next thing to really think about is lifestyle changes. So, we've talked a little bit already about nutrition and getting all of those key nutrients, but other factors in your lifestyle have a huge impact on methylation as well. So, something has to have been out of balance to have gotten an autoimmune disease. Methylation just doesn't stop working well for no reason. And this is something that I cover these different lifestyle factors in my book and how to really optimize them. I think it's fairly comprehensive, and there are plenty of other books out there that cover these same lifestyle factors as well in detail. So whether it's my book or someone else's, please educate yourself and, have a look at what you can do to really make some lifestyle changes that are going to support your methylation. Um, we've also got flow-on effects of methylation issues. So this is kind of a little bit more obscure or abstract, but I don't want to go into a lot of detail on it, but I just wanted to mention it so that you're aware. And so 100% of the clients that I work with have some sort of heavy metal toxicity. Some of these are people with autoimmune diseases. Some of them have got other health concerns as well. But there is a growing body of research linking things like heavy metal toxicity and pathogens at the site of autoimmune attacks. So, what do I mean by that? In Hashimoto's thyroiditis, we know that heavy metals like mercury, nickel, and cobalt can actually directly interfere with thyroid function. So, these are endocrine disruptors and they bind to thyroid hormone receptors. So, they accumulate in the thyroid. And then what happens? We end up with methylation not working well there, and you end up with an antibody attack there. Same sort of thing can happen with, say, Crohn's and MS, but with pathogens. It's the site where those pathogens are, is where the antibody attack is actually happening. And poor methylation is linked to both of these. Now, the last thing I just wanted to say quickly is that you can actually support your methylation by treating yourself with kindness. And I think that anyone who's had an autoimmune disease will know what I mean when I say that, I've had people say to me that you're not really sick or I can't see anything wrong with you. And they think that you're a hypochondriac. I want you to know that you're not. I see you. I know exactly what you're going through. It's real. You are exhausted, and that's okay. You can actually slow down. You can do a little bit less than you used to do. You can do a little bit less than other people. You can give yourself a break and actually try and recover from this. And that actually giving yourself a break is going to be key to that recovery. You need to do that so that your body can catch up and just get a bit of a breath. So that's kind of where I wanted to end. I feel like that was really brief because there's so much more I could have shared about methylation.